All right, welcome. So today we're going to be talking about cancer genomics, and we've moved the FEWAS lecture to Tuesday, followed by the genome engineering lecture on Thursday. So again, this is part of Module 6, the more advanced current research directions. So the goal is to sort of understand how do all the technologies that we've learned about throughout the term help us fight one of the most challenging health issues of today, namely the huge diversity of cancer diseases. So first we're going to talk about basically what defines cancer, what are some of the hallmarks of cancer, and how do we understand the function of cancer. Then we're going to look at how we can tackle understanding cancer through whole exome sequencing, through whole genome sequencing, and through understanding the functional genomics of cancer, and specifically epigenomics. So we're going to look at drivers at the protein coding level, drivers at the non-coding level, and also epi-alleles and drivers at the genomic level. And then we're going to focus on an area of great current interest, namely tumor immunology and all of the recent immunotherapies that have been developed. And this is truly revolutionizing the way that we understand cancer therapies today. So back in 2000, Bob Weinberg and Douglas Hanahan were asked to write a review for Cell, basically summarizing, the latest and greatest of current understanding of cancer. And they came up with this idea of the hallmarks of cancer. And then the idea was that the cancer, through its progression, needs to acquire a certain set of skills. It needs to learn how to grow. It needs to avoid dying. It needs to bring together enough blood supply to feed it and feed its growth and its energy needs. It needs to be able to replicate indefinitely, whereas most of our cells only replicate a finite number of times. It needs to metastasize. It needs to actually invade new tissues. And it needs to ignore the signals from the body that normally stop cells from dividing uncontrollably. So putting all this together, he basically went through the entire body of literature at the time and basically said, well, all of these different signatures of cancer can be classified as to one of those 6 different categories. And that actually became a very important way of thinking about cancer through the early 2000s. To the point that in 2011, the same authors were actually asked to write a revised review of the new hallmarks of cancer. And that actually tells you a little bit about sort of what changed in that decade. And what you'll notice is that, for example, avoiding immune destruction was not even on the original list, even though cancer immunotherapy is in fact one of the most prevalent aspects of cancer biology nowadays. Similarly, tumor promoting inflammation. So basically this is taking advantage of the, not the adaptive, but the innate immune system to actually help the cancer by actually bringing in blood and other signals. Energetics is in a way expanding this whole concept of angiogenesis, but actually feeding the cancer with a lot of nutrients and a lot of energy, coupled with angiogenesis. And of course, one of the most important hallmarks is the concept of genome instability. The concept that the tumor, in fact, promotes mutations that make it more mutable. And these kinds of mutations are very detrimental to most cells. But when you're a tumor cell and you're dividing uncontrollably, then you can work the numbers to your advantage. So the more mutagenic you are in a way, the more it helps you explore different evolutionary paths that can then allow you to evade the immune system and adapt and evade therapies and so forth. Everybody with me so far? So then the idea was that you would acquire each of these, a huge number of mutations, but then you would need to acquire mutations that enable each of these different capabilities in some order. And it's only when you acquired all of them that you would then become, potent cancerous cell. Okay? And there can be many, many different pathways to achieve this self-sufficiency of the tumor. How is that done mechanistically? This is done in a major way through mutations, but not exclusively through mutations. And what's really interesting here is to recognize the diversity of mutation paths, if you wish. So we are all born with common variants that have been going through the population for many, many generations. In addition to those germline variants that are very common, we have germline variants that we inherited from our recent ancestors, where one of these mutations may have happened, in my great-great-great-grandpa. And it's not common in the population, but it's common in my family. And then in addition to these germline mutations that I inherit from my parents and their parents and so on and so forth, there's a large number of additional mutations that happen somatically. So we talked about brain development a couple of lectures ago, and how, in fact, on Tuesday, and how we can use single-cell RNA-seq, for example, to trace mutations that happen somatically during the development of the brain, and that some of these mutations are, in fact, not under purifying selection, and therefore they're allowed to have much stronger effects. In the case of cancer, this flips the equation completely, and it says not only are they not selected against, because cancer cells expand, there's an evolutionary process which might actually favor these mutations, these strong effect mutations. And therefore, you might actually have, a preponderance of these somatic alterations with strong effects. Is everybody with me so far? These mutational changes, both inherited and during the somatic expansion of the body, can then lead to stable or transient changes in gene expression, in epigenomic alterations, in regulatory networks, and that can then lead to a self-sustaining state that then facilitates additional mutations and additional growth. And then, of course, all of that can be detected through very different approaches. On one hand, sequencing and profiling and epigenomics and transcriptomics. On the other hand, looking at histopathology, looking at different blood biomarkers or imaging biomarkers, and sort of the progression of that. And that ends up with many different cancers coexisting in the body of a particular person. So basically, cancer can be thought of as a combination of germline mutations, somatic mutations, epigenomic changes, and gene regulatory alterations that can then give rise to different lineages that are then co-evolving and altering and progressing through the body of a patient. The important thing to realize is that the vast majority of these mutations are meaningless. They don't tell you anything about cancer. It's only a tiny little subset of these mutations that are actually the driver mutations. All of the other ones are, in fact, passenger mutations. They just happen to tag along. And it's very important to differentiate between driver and passenger mutations. The driver mutations are the ones that confer the advantage to the growth of the tumor. And then the passenger mutations do not directly contribute to the fitness of a tumor, but can be used to, for example, trace the lineages of the tumor and so on and so forth. And of course, distinguishing driver from passenger is extremely challenging. And that's sort of part of the needle in a haystack problem of having many different candidates, only a very small number of which will in fact be driver mutations. Okay? You basically start with some intrinsic mutational process, and then there's additional exposures that result in additional phenotypic changes. And then once you have a mutator, you can then start accumulating many mutations. And then you should also realize that during any form of therapy, there's continued mutations that can then select specific clones of the tumor and expand those in response to the therapy. So it's a moving target. It's not just, hey, whatever the cancer has accumulated until that point. It's also the fact that in response to the therapy, there are additional mutations. Yeah. Very, very good point. And that's in a couple of slides. Yeah. So then the question is, how do you distinguish drivers from passengers? The key insight from genetics of common variants is the whole concept of, hey, if it precedes the disease, then it must be causal. Basically, the ones that are inherited and associated, if you find a correlation between a genetic mutation from a germline and a phenotypic variation, then you can sort of infer causality. For the somatic mutations, this is much harder because many of them might actually be a consequence of the tumor, or it might be a consequence of treatment. They're not necessarily drivers. So to distinguish the drivers in that case, what we're looking for is recurrence. Basically, the concept that that gene is mutated in multiple clones within the same person or in multiple patients of the same tumor type and so on and so forth. So the paradigm there is not one of causality because of temporal precedence, but one of a recurrence compared to what you would expect by chance. Any other questions? All right, so there's multiple different types of mutations. We can categorize them into broad classes. We can categorize them in terms of proto-oncogenes and oncogenes. These are genes that promote normal cell growth, and when mutated, they stimulate overgrowth. So they're basically leading to overactive replication. Tumor-suppressor genes are the brakes. So these are the gas pedals, and these are the brakes. These are genes that normally slow down cell division. And when mutated in a cancer setting, they exhibit loss of function, and that allows for unchecked cell growth. So there's two types of things that drive the cancer. On one hand, it's sticking the accelerator down, and on the other hand is like, getting rid of the brakes. Okay? So that's the difference between oncogenes and tumor suppressors. Then there's a whole other class of mutator genes. So these are genes that simply increase mutation, and that basically allows you to have many more opportunities to have one of these, step on the gas or get rid of the brakes, additional mutations. And then there are additional mutations that can happen that can then dysregulate the circuitry of the cell. Even if they don't change the DNA directly, they might actually simply, overexpress a particular pathway, lead to a stable epigenomic state, for example, a pluripotent state from a terminally differentiated cell. Basically, if it de-differentiates and gets stuck in one of those, highly pluripotent, highly self-propagating state, then you can actually land in a stable state, even if you don't have a mutation specifically. So it's important to recognize that some of these gene regulatory changes can themselves be stable. Everybody with me? So here's one example of a tumor suppressor, so P53, also known as tumor protein 53. Serves as a tumor suppressor, and it's commonly known as the guardian of the genome. It serves a key link between DNA damage and repair. And P53 is by far the most mutated gene in many different cancers. And its mutations basically cause loss of function. And then it enables, you've basically gotten rid of the brakes, it enables tumor emergence and tumor growth. There's many different pathways that can lead to the function of P53 in terms of growth arrest, apoptosis, angiogenesis prevention, and so forth, which then, when mutated, leads to just dramatic havoc in the gene. Okay? Basically, normally P53 leads to cell cycle arrest if it detects a mutation, and then repair, and only then is the cell cycle allowed to restart. Or if you're not able to repair, you just simply undergo programmed cell death, known as apoptosis. Whereas in a mutated P53, despite DNA damage, you're allowed to continue the cell cycle. Okay. Here's a converse example. So the RAS family proteins, they're members of small GTPases that are involved in cell growth and cell cycle. And then mutations in the RAS family can basically cause overgrowth and overproliferation. So this is an example of a tumor suppressor. That's an example of a proto-oncogene / oncogene. Basically, RAS mutations can then lead to all kinds of activation of proliferation rather than apoptosis or death control. And these mutations then lead to all kinds of downstream pathways that affect many, many different aspects of the cell, including cytoskeleton, survival, messaging, endocytosis, gene expression, cell cycle, motility, apoptosis, and so on. Another very common type of event is when you bring together the regulatory circuitry of one gene and the protein of another gene. So you can basically create these chimeric genes where you can have either the first many amino acids of one and then the next many amino acids of the other, or the regulatory circuitry of one and the gene body of the other. And then that allows you to sort of create new functions that are normally not there. And then there's some famous examples that basically drive different types of cancer by creating these chimeric proteins. And then, there's many different types of traits where this is found. Recognizing that these hallmarks has actually been a very important driving principle in sort of targeting some of the therapeutics against cancer. And one of the leading paradigms of that is the concept of addiction, where the cancer becomes addicted to a particular oncogene. And then, even though most cells don't depend on that particular gene, this in a way becomes an Achilles heel for the cancer because it has rewired its circuitry to be so dependent on some of these hallmarks. You can then say, well, I can just go and maybe target specifically these oncogenes and then have a much stronger effect on the tumor. Whereas for normal cells, they will not be as affected by this type of mutation. So basically, a tumor cell that has now become dependent on a particular function, when you mutate that function now, it can, actually impede the growth of the tumor. Everybody with me so far? And then these mutator genes that I mentioned are basically genes that simply lead to lower repair efficiency. So as you know, with every cell division, the genome basically takes great care to detect mistakes and then go and repair them. And then if these pathways of repair are broken, that in fact increases the mutational rate of the tumor. And that allows you to, explore much more of that potential space. So there's many mutator genes that are involved in DNA repair pathways, in controlling chromatin stability and movement during different phases of the cell cycle. And then when you mess that up, that can then lead to, really havoc in the genome. Many of these cells will die, but then some of these cells will basically become, much more potent. Yep. Yeah, so basically, this is not a meiosis phenomenon anymore. This is really about, oh, I see. Like during M phase versus interphase. This can happen at any point on the cell cycle. And then, as I mentioned earlier, so you can actually have these chimeric proteins, which are made by, for example, deleting a big segment between two consecutive proteins, and then you now have a fused protein, or by sort of creating rearrangement. And these are some of the examples of the different types of rearrangements that you can have. You can have an inversion, you can have translocation, you can have duplications, you can have tandem duplications, you can have deletions. And all of these can basically lead to, just creation of new functions that can lead to, cancer proliferation. So one such example is polyploidy, We talked about whole genome duplication a few lectures ago, and then how that actually enables the evolution of new pathways, because you've now duplicated the entire genome. So then you can start losing genes and then creating new functions for genes, amplifying entire pathways, and so on and so forth. Aneuploidy is a gain or a loss of an entire chromosome or a chromosome arm, and then chromosomal duplications and deletions. And then these can lead to dosage effects where some genes are expressed much more, and gene regulatory changes where genes can now be in a new context. Okay? So this is basically just introducing the foundations of sort of how to think about cancer and what are the different events that can lead to cancer. Who feels that they've learned something so far? Awesome, good. Any questions? Okay, so now let's basically focus on how do we go about actually using genomics to understand the function of cancer. So one of the main strategies has been to look at both common variants associated with different types of cancer that are inherited, as well as exonic variants from whole genome sequencing of the tumors themselves. So how do we identify cancer driver genes and driver mutations? And that's, again, the key goal of exome and whole genome profiling. And then it's important to recognize that mutations can be across the entire spectrum of common variants, rare variants, and somatic variants. And then, in fact, this is exactly how we ended up with the hallmarks of cancer. So, a few decades ago, we had no idea how cancer actually functions. But then by genome sequencing and genome-wide association studies, we are now able to recognize what are the genes that are repeatedly mutated. And that basically led to the realization that, wait, these genes are following a small number of common pathways. There's 3 types of mutations, and these are coupled with different types of analysis. So genome-wide association studies are basically focused on usually weaker effect, non-coding mutations, which are common in the population, whereas linkage analysis within a family can basically lead to the discovery of strong effect Mendelian mutations, which are different from family to family, but then very often converging onto the same gene being mutated, just in different ways in each family. And then with whole exome sequencing or whole genome sequencing, the goal there is to find strong effect somatic mutations that arise during mitotic cell division. So if you look at the progression of how that tumor came to be, you basically have a genome that was inherited from the lineage of that person, and you basically have some ancient mutations that are very common in the population, some recent mutations, maybe in the last few generations, and then eventually a fertilization event. And from that, you end up with a highly mutagenic process because of the huge number of divisions that happen. And that can basically lead to accumulation of passenger mutations as well as driver mutations. And the driver mutations in isolation might not be selected for. But as they start accumulating more and more of these hallmarks, you then start getting an expansion. And then, for example, you might have a clonal expansion. So this particular tumor may have accumulated a sufficient number of these driver events that it can now start proliferating, metastasizing, recruiting blood supply, evading the immune system, and so on and so forth. So basically, when you're looking at a tumor at some endpoint, you need to recognize that all of the mutations that it has are in fact accumulation of all of these previous events all the way to the tumor. Okay. So the challenge, of course, is that there's a huge imbalance between a very large number of passenger events and a very small number of driver events. There's only 5 to 20 driver events compared to thousands of passenger events with roughly 1 mutation every megabase. So that basically means that it's very difficult to recognize the true drivers when we do this kind of analysis. So let's go through them. So basically, number 1, common variants. So you've seen these types of plots before. Here I'm only showing cancer-associated mutations. Basically common variants. These are ancestral mutations, common polymorphisms now. And there's hundreds of genome-wide association hits for many different diverse cancer types. This allows you to now do polygenic risk prediction, but these effects are usually weaker, and there's no Mendelian inheritance because these happen in the context of many, many other mutations. It's really the combination of these mutations that predispose a particular person or a particular family into different types of cancer. Okay. Then you basically have the ability to trace which segment of the genome was inherited by the patients showing cancer. So basically, in different lineages across, in this particular case, 4 generations, you can basically see who were the individuals that actually had developed cancer. And then you can ask which segments of the genome do they carry. And with every additional person that you have, you basically can exclude half of the genome roughly, because these relatives are just 50% identical to each other. So that means that very quickly you start sort of narrowing down the fraction of the genome that could be explaining a particular cancer predisposition in that family. And then these mutations have much stronger effects. So you can actually now trace them as Mendelian inheritance. And when you do that across many, many different families, what you realize is that the same genes are in fact mutated in different families. And this is exactly how Mary Claire King, in fact, mapped one of the most famous breast cancer genes, BRCA1, on this arm of chromosome 17 back in 1990, using genetic linkage analysis in families that had an inherited risk for breast cancer. And what you notice is that every family carried a different set of inherited mutations. Why not a common set? Because every single time one of these mutations rises in frequency, selection is acting against it. So any one of these mutations only survives so many generations before its frequency drops in the population, but new mutations arise, and these are sort of what leads to this Mendelian inheritance in family. Everybody with me so far? And then the last part is, of course, somatic mutations. So, that's where a lot of exome sequencing has been focusing on, and more recently called genome sequencing. And then the concept here is that we're going to be sequencing both the normal genome for that person, basically taking non-cancer cells from that person, and the tumor genome from that person, by basically going into tumor tissue and sequencing the genome there. And then the goal is to recognize recurrent mutations, basically mutations that happen in many, many individuals, and that these are then leading to hotspots that can then be implicated in cancer. What you should note is that because these mutations arise only later in the order of cell divisions, they will not be present in 100% of the tumor. So when you sequence the tumor, you might end up with only 10% of the tumor actually showing these mutations, or 20% of the tumor. So what you need is very highly sensitive methods for actually calling mutations, because only a small fraction of your sequencing reads will show the mutation. So depending on when the mutation arises, you can basically, cover only a small fraction of the tumor. So that basically leads to these very low allelic fractions. Does that make sense? So basically, if a mutation happened early in the tumor, it will be at 100%. But if a mutation happened later, it might only be at 10%. And if your base calling methods basically say, oh, I'm just going to call the majority base at every position, you're going to look at the tumor and say, oh, well, 90% says that there's no mutation here. But that's because the driver mutation was only at 10% frequency. Who's with me on that concept? Great. So then, once you've identified the mutation, you basically need to see if is it recurrent? And that might sort of, basically when you first look for these mutations that are specific to the tumor, you will end up with a huge, huge number of passenger mutations and only a small number of driver mutations. And then you can apply different filters. You can basically say, hey, is it recurrent? And that can eliminate, for example, many of the passenger mutations, but also some of the driver mutations. Is there, do my computational methods predict that there's a functional impact? And then that can sort of weed it further. Do I find that it is in fact a recurrence across, different tumors? And then that can perhaps exclude most of the passenger mutations, but then keep most of the driver mutations. And then you can go off and experimentally and functionally validate it, hopefully ending by the end of that process with mostly true driver mutations. Everybody with me? So there's many computational pipelines for doing this. Here's one such pipeline developed by Cathy Goetz's group here at the Broad Institute for calling somatic point mutations, so MUTECT, for mutational detection, basically. So this is a Bayesian classifier that basically determines if the mutation is tumor-specific using paired sequencing of both tumor and normal from the same individual. And that allows you to basically recognize these mutations, even if they're only at 10% frequency. So, you can basically sequence the tumor, sequence the normal, and if you find a mutation that is repeatedly found in the tumor, even if it does not cover all of the reads that you get from that tumor, you can still sort of develop computational model that basically tells you, well, how much more likely am I to see this by, if it's a true tumor versus simply by chance. And then you can apply various filters. For example, is it near a gap in the genome assembly? Is there a particular bias in the strand? Do I find it mostly on the right, on the sort of forward reads versus the reverse reads? And that can allow you to reject sequencing errors that are associated with specific technologies of sequencing. Do I find it in, poor mapping regions? Do I find triallelic regions where you basically don't just have two alleles, but multiple alleles? Is it clustered in particular position on the reads? And is it also found in the control? So basically, when you sequence both tumor and normal, you can basically say, well, hey, this is also found quite frequently in normal, and I'm gonna be more likely to exclude it. And then you do that across many individuals, and then you classify the variants and start calling variants that way. So there's also pipelines for annotating the somatic mutations. So basically using all of the different resources that we talked about in class in terms of common variants from the 1000 Genomes Project, rare variants from ClinVar and other projects, known protein coding altering variants that basically whose function is easily predicted, and also just the gen code annotation of protein genes and catalog of DNA repair genes, understanding many different properties that one should correct for. So once you have your mutation, you can basically annotate them, prioritize them, and you can, then start looking for recurrence. So basically going down that list of enriching for tumor mutations and then eliminating the passenger mutations. The concept of recurrence can be applied at many different levels. You can be recurrent at the nucleotide level, where the same amino acid is mutated, recurrently across different families. You can be recurrent at the gene level, where you can have multiple deactivations of the same gene. So this is one example of BRCA1 and BRCA2, where there's just a huge number of different deactivations of that gene that can happen, each with different degrees of severity. So BRCA1 and BRCA2 are very recurrently mutated, and each of the individual's mutations is also recurrent. So, there's more than 300 mutations for BRCA2, more than 500 mutations for BRCA1, and now that a lot more genome sequencing is going on, we're basically increasing these numbers dramatically. Everybody with me so far? So the decrease in sequencing costs has basically led to very many projects for large-scale cancer genome sequencing and cancer exome sequencing. And that has basically revealed many genomic hotspots. So by basically analyzing the mutation profiles across tens of thousands of tumors, across many different cell types, the community has now called more than 2 million somatic hypermutations, with an average of 57 mutations within the exome of a given tumor, affecting many different genes and clustering into a small number of hotspots into a relatively smaller number of protein coding genes. You can see here just a completely unbiased view of sort of what are the cancer driver genes, both in terms of tumor suppressors as well as oncogenes. And you can see here that at the very top is TP53 that we talked about earlier. And then there's many sort of usual suspects that are recurrently found. Everybody with me so far? So basically, there's two sides of cancer. On one hand, you have recurrence, and on the other hand, you have heterogeneity. So basically, recurrence is the concept that a small set of pathways are altered, and these are necessary for cancer. This is the concept of the hallmarks of cancer. And then oncogenes and tumor suppressors are very common points of recurrence. And at the same time, you have heterogeneity. Basically, cancer is an evolutionary process which is driven by positive selection. There's a very large number of precancer cells, and these are constantly subjected to selection. And there are many, many different ways that an oncogenic pathway can be hit. So you have this huge, huge number of paths that are constantly being explored. And if some of them accumulate more mutations that make them more virulent, more pathogenic, then they can start spreading more. So to understand that, we can basically start looking at the process of clone expansion. And you can think of it in this particular fashion. You basically have, just a normal cell, and then it has now acquired a number of mutations that make it, act like a tumor. And then these cells are now expanding at some rate. And then one of these cells, only one of these cells, may acquire an additional mutation that now make it divide perhaps faster. And you can then sort of see that clone expanding and taking over the original clone. And you may have another new clone starting from perhaps the normal lineage here. And then it will start expanding, but maybe it will be taken over by the huge expansion of this red clone here. And then there's an additional mutation that happens to one of these cells, which leads it to expand even further, and so on and so forth. So what you end up with is an evolutionary process that tells you just at each time point, what are the mutations that are still in the running. And when you end up with a tumor, you're only looking at the end of the selective process. And, when you're looking at a metastatic event, it might not even represent the latest of that lineage. It may have actually metastasized earlier from yet another clone. So that's sort of why understanding these evolutionary processes within the body is actually extremely important. Everybody with me so far? So genomic instability can be a source of this functional heterogeneity. You basically have a large number of abnormalities that can lead to instability, that can lead to more mutations, that can lead to more diversity, and then more functional heterogeneity. And instability can be detrimental to individual tumor cells, but as a whole to cancer, it is sort of what lets it survive, because some of these cells will actually escape the bottlenecks and then increase the number of paths to cancer. So, you basically might have some kind of bottleneck here over time, and only one of these cells might have a sufficient number of mutations or a particular mutation that was missing from the other ones that allow it to then further expand. So again, the tumor plays a numbers game. There's very little purifying selection, and therefore you can just decrease the numbers and get at that. And then tumor heterogeneity is a feature of both the emergence of a tumor, as well as the resistance to treatment. So the more heterogeneous the tumor is, the more likely it is to evade treatment, because some of these cells might actually develop a sufficient number of novel mutations that can then now allow it to further regrow. You can basically see here the tumor coming under control with some treatment, and then one of these cells accumulates a new type of mutation that allows it to expand further, and then additional clones within them, and then perhaps a metastatic event, and so on and so forth. And that's why even after chemotherapy, very often you basically have these tumors coming back. Then we can use that to start tracing the clonal history of multiple metastatic sites. So for example, sequencing both metastatic lesions and primary lesions across different locations in cancer patients. And then what they found is that the mutations can be regionally distributed and partitioned into private, shared, and ubiquitous, where basically different clones are in fact sharing different subsets of these mutations. So some mutations are ubiquitous, others are shared in the primary tumor, others are shared in the metastatic tumor, and others are purely private, basically. Then they can infer the phylogenetic relationships between the different tumor regions and recognize differences in ploiding. You can actually start building trees of how these regions are changing. And you can build computational models that allow you to now trace these events. So you can basically build probabilistic models that explain sort of what are the different sources of heterogeneity, what are the different mutations, and then measure across time and space to infer that phylogenetic history. So this is one example of a patient. This is a collaboration that our lab has with Geneviève Poland. This is basically looking at a single patient who underwent treatment over many different types of drugs. So one type of drug, another type of drug, another type of drug, and so on and so forth. And you can see here the days after treatment. And some of these are 720 days, 1,000 days, and so on and so forth. So this patient has basically been profiled for more than 3 years. And now we can go back and start asking for different biopsies across different locations in their body. How are these biopsies related to each other? And what we're finding is that we can build phylogenetic trees, basically telling us how are these mutations related to each other, how are they growing, and we can recognize at least 5 distinct lineages that are co-evolving within the same patient. So it's not true to think that sort of these clonal expansions completely abolish the other lineages. In fact, you should think of it as a co-evolutionary event of multiple lineages coexisting within the same patient. And what you can also see is recurrent events. For example, there's a genome doubling that happened here on the green lineage, and there's a different genome doubling that happened in this light blue lineage. There's different mutations that can be sort of recurrent, and that allows you to sort of learn even in the context of a single person the strategies that the tumor is undertaking to evade therapy and to recur. Everybody with me so far? So we talked about exome sequencing, about recurrence, and about these evolutionary dynamics of these clonal expansions and these common signatures. So this has basically focused at the gene level, but then the tumors are so much more complex. So whole genome sequencing has now allowed us to go beyond just the protein coding mutations to start understanding also non-coding drivers. So in this particular case, you can basically do whole genome sequencing and basically just ask where are these mutations happening. And what you're finding is that the vast majority of these somatic mutations are in fact non-coding. And of course, most of them will be passenger mutations. They're finding the drivers will be hard, and we can start making sense of these non-coding mutations by using these principles of recurrence. And this is similar to coding, but now the boundaries are not known. The background mutation rates are not known. And they vary by region, by patient, by state, by chromatin state, by DNA location. And convergence is basically now a little harder because you don't have sort of this concept of a gene where things are converging. So these are scattered mutations, which might be hitting different enhancers. And only when you map these enhancers to the same target gene will you then be able to recognize the recurrence event. So there's basically a series of pipelines. This is one example by Ekta Kurana from Mark Erstein's lab, who's now leading her own lab for basically detecting and interpreting these non-coding mutations. So basically you can count the number of events that are happening relative to a background. You can basically build statistical models for enrichment and calling of drivers through recurrence. And then you can sort of functionally annotate each of those by integrating with ENCODE and Roadmap and other databases. Basically functionally prioritize the ones that are in accessible regions or the ones that are perturbing regulatory motifs. And then you can go in and experimentally validate some of them. So when we do these kinds of sequencing studies, the first thing that you'll notice is that there's dramatic changes in the background average mutation rate of different types of tumors. If you look at melanoma, for example, you have 100 times more mutations than if you look at, say, thyroid cancer. So different types of tumors are hugely different in terms of their average mutation rate. But the other thing to realize is that within a particular tumor class, there can be, again, orders of magnitude difference in the mutation of that tumor. So basically, across different patients that all have melanoma, you can have, 100-fold difference in the mutations themselves for each patient. So, that's the first part. The second part is to recognize that different tumor types are in fact showing very, very different types of mutations. So, for example, melanoma is here dominated by C2T mutations, whereas these lung adenocarcinomas and squamous cell carcinomas are in fact showing a huge prevalence of C2A mutations. And this is due to the specific mutagenesis profile of cigarettes. So basically, they're a mutagen, and they have a different signature. And if you look at bladder cancer, stomach cancer, or ovarian cancer, and so on and so forth, you basically find very different prevalences of these signatures. And that's actually very, very important because you can actually use those to recognize different types of cancer, even in a metastasis setting. You can basically say, well, this cancer is found here, but in fact, it has a signature of a different type of tumor. Yeah. So the way to think about it is that there are different processes that act in different parts of our body and different cell types within the same tissue. So, for example, the repair machinery of your liver might be different than the repair machinery of your brain. So basically, the specific set of proteins that are involved in that repair machinery might actually be, tilted towards one type of, mutation and one, detection and repair versus another type. And depending on the specific proteins that are the most active in, for example, that repair machinery in different cell types and different tissues, you might end up with a different signature. So there's hundreds of proteins that are involved in DNA repair, in sort of recognizing the sites and fixing them and so on and so forth. And a different subset is in fact active in different cells, and a different subset is mutated in different tumors. And that's sort of one mechanism that leads to those differences. So, for example, your skin is exposed to UV radiation. Your lungs are exposed to smoking. The mutagenesis itself might actually be different. So basically, depending on the region of the body, you might be exposed to different types of mutagens. If you look in your lung, if you look in your liver, if you look at your intestine, the local community of bacteria living there, for example, that is also affecting the types of mutations that can actually, be undertaken by different cells in the body. So basically, there's two factors here. On one hand, the repair machinery itself can vary from cell type to cell type and from tissue to tissue. On the other hand, the types of exposures can vary from tissue to tissue. Does that answer your question? Any other questions? All right, so on one hand, basically understanding these background mutations is extremely important. The background mutation rate is extremely important. Second, understanding specific types of mutations is extremely important, because now when you find a particular type of mutation, you can say, well, what would I have expected for that particular tumor type? So again, these reflect differences in mutator genes. Different repair genes are most active in different tissues, but also the exposure to different mutagens is active. So that's part of the reason we have these different mutational profiles. If you now look across the genome, you basically also find that there's great variation in the mutation rate of different regions. So If you look at mutation rate here in red, basically see these, tenfold differences in mutation rate. And if you look at gene expression levels, you basically find that, hey, there's a correlation between them. And if you look at the time of replication in the cell cycle, you also find that there's a correlation between them. So what's going on here? You basically have, these different, seemingly completely unrelated processes that are obviously correlated. So one way to interpret that is that there's transcription-associated repair. So basically, wherever you transcribe genes, you basically bring the repair machinery along. You should also realize that as you open up the DNA for transcription factors to bind, so does the repair machinery. So in regions of the DNA that are very close, the repair machinery is simply not accessing these regions. So if you have tightly packaged chromatin and you have a mutagenesis event, like from UV radiation or from smoking, you can basically have these mutations happen but go unrepaired. But, depending on where these mutations happen in the genome, the probability of actually repairing them can be very high, and also the probability of mutation itself can vary greatly. The other thing to realize is that there are specific classes of genes that have simply a lot more mutations. So basically, in the early studies of whole genome sequencing, the genes that kept coming up with the most mutations were in fact olfactory receptors. We talked about olfactory receptors a few lectures ago about how humans have basically sort of stopped worrying about them so much. And these obviously are not cancer drivers, but they're still highly mutated. But when you adjust for this background mutation rate, what you realize is that they actually fall out. So, the ability to distinguish between, tumor and normal, between how mutagenic is every particular region and how many mutations it accumulates overall, what you realize that genes that are both long and are devoid of selection will basically end up having more mutations to start with. So being able to correct for that background mutation rate is in fact what starts leading to some of the cleaner signal. Everybody with me so far? And as I mentioned earlier, mutations greatly correlate with DNA accessibility. Inversely correlate with DNA accessibility. And that's because the repair machinery is actually optimized for accessible sites, which would otherwise become hypermutated. And in cancer, the repair machinery is often disrupted. The cancer mutations are enriched for DNA sensitive sites compared to what you would expect. It's all of the above. So basically, if you look at the specific event, C2T is in fact the amination. So, it's not a surprise that this is leading, but all the other ones cannot be explained that way. So, very good. Mm-hmm. Yeah, but that's exactly what these methods are doing. They're basically constantly matching regions and then correcting for that. So basically, you're correcting for background mutation rate of the entire cell. You're correcting for the mutation rate in specific cancer type. You're correcting for the regional mutation rate. Oh, sorry. Say it again. I think after you correct for that, the frequency of driver mutations is so low, you don't have these, dramatic variation. They then start clustering in really functional classes, more closely related to the whole. So you're correcting for these, at the background mutation rate, at the cancer type mutation rate, at the regional mutation rates, at the accessibility mutation rate, and you can also look at the specific chromatin state. So this is a collaboration with Richard Salari, where he basically looked at different types of histomodification marks, different chromatin states called from them, and then define promoters, enhancers, general regulatory, poised, repressed, low abundance, open chromatin, and so on and so forth. And what you found is that the mutation rate varied greatly by chromatin state as well, giving you yet another layer of complexity. And the mutation rate also varied greatly by patients. So basically, if you looked at different patients, you basically found that there was this dramatic correlation with the patient. You could order the patients based on their overall mutability, and that actually led to increased mutation in each of them. So putting all this together, you can basically develop methods for correcting for the expected number of mutations and then calling the excess mutations for each of them. And then NCD Detect is one of these programs. So this is again in collaboration between our lab and Marlena Rasmussen, who's in Jacob Pedersen's lab in Europe. And that allows you to basically detect non-coding driver mutations by accounting for all of these different sources of background mutation rate. And once you've done all that, you can then start functionalizing these mutations by basically asking, hey, where are they falling? And you can basically start recognizing motif gain, motif loss, change in the transcription factor binding, rewiring of different regulatory regions by basically bringing them together, post-transcription regulation by basically creating or abolishing microRNA binding sites, as well as sponge events where you can basically overexpress a particular RNA, which will then bring with it a bunch of transcription factors or a bunch of microRNAs that are now no longer going to their target sites, enabling you to sort of, have these global changes in activity. So again, Richard Salari used all this to now develop a convergence model that allows you to go from distal enhancer regions to their target genes and ultimately common pathways by exploiting high C links in the chromatin and basically bringing together these very scattered events across patients into high frequency recurrent events when you sum up across all of the different regulatory regions of the same target gene, giving you sort of the recognition of true targets from otherwise scattered and unremarkable mutations. So then using that, he's able to predict dysregulation of specific genes as driver events in the same tumor type, even though you have to add across patients and across different enhancers, these are all converging onto the same target gene. Basically, he's building these regulatory plexus for every gene, mapping non-coding driver mutations onto the plexus, and then recognizing driver target genes, even in absence of any protein altering mutations. Then you can experimentally validate these non-coding drivers by sort of building enhancer reporters to show that these mutations are indeed changing the activity of the genes, and then to show that these genes are indeed having a function in a functional setting. So this is now looking at exonic as well as whole genome sequencing. And again, the types of questions are different and the types of analysis are different, but they're always getting at these recurrent events. Basically correcting for the background mutation rate across regions, across chromatin states, across patients, across the regulatory plexus, and then looking at convergence of many mutations into the same enhancer, many enhancers to the same gene, many genes onto the same pathway. I'll speak briefly about sort of another class of events beyond these mutational events, which are epigenomic alterations. So basically more and more, we're recognizing that many of these mutations are in fact affecting the regulatory machinery of the cells, and specifically master epigenomic regulators, which result in global epigenome-wide changes. So this seems counterintuitive at first. How can you get a very specific response in a particular pathway through these global mutations? And then the answer there is that because cancer is playing the game of the numbers, it can exploit these global dysregulatory events because one or more of these targets will basically lead to something that's advantageous to the cancer. Very often, these are changes that are leading to sort of de-differentiation, so basically a return to a more pluripotent state, And then these can be, manifested in many different types of events. For example, through global activation by loss of DNA methylation, global repression by gain of DNA methylation, global repression by gain of repressive histone mutations, or just state changes through epigenomic remodeling and nucleosome positioning. So these can vary by cancer type, and these are, very often leading to the recognition that many of these master regulators are in fact putative target genes for cancer therapy. In fact, there's several companies that have been started in that space, including one by Sigal Kato, also another member of the Broad Institute here. So this epigenetic reprogramming can basically become a guide for specific therapeutics. You can basically diagnose cancer as altered differentiation and then treat by reprogramming the cancer cells towards normal development. And one approach for doing that is to basically do single cell RNA sequencing, as we talked about on Tuesday, and then start recognizing the different programs that these cancer cells are undergoing, and then sort of, recognize what are the dysregulatory events and how can we fix them by sort of reversing these changes. And this has basically led to a lot of recognition of the heterogeneity in the tumor microenvironment, basically looking at the interaction of that tumor with its environment, specifically the immune environment in its vicinity. And then that can lead to a lot of heterogeneity in where you are in the cell cycle, what is the state of each cell. And then there's a lot of methods for matching both single cell DNA sequencing and single cell RNA sequencing data to infer clonality events through single cell level. The last topic is something that has dramatically changed the face of cancer treatments in the last decade. So this is basically understanding the interplay between the tumor and the immune system. So as I mentioned earlier, the tumor interacts very heavily with its microenvironment. So the microenvironment can be defined as sort of the local context of cellular and intercellular agents that are surrounding the tumor in its position in the body. So every tumor has a variety of cells in its microenvironment that provide key interactions that either repress or promote the growth of that tumor. You can have basically cancer stem cells, you can have cancer cells, you can have cancer-associated fibroblasts, you can have endothelial cells, pericytes, and a lot of immune and inflammatory cells that are coexisting in that environment. So then understanding the interactions between these cells is extremely important, because you can actually start helping the body fight the tumor rather than sort of fight it in a completely separate way. And the key recognition here, and that's something that only happened in the last few years, is that the tumor is in fact rewiring its own immune profile. So basically, very early in tumor emergence, the tumor evades the adaptive immune system by altering its own immune profile. This is basically simply Darwinian evolution. Tumor cells that are recognized are killed off, and over multiple generations, this leads to a selection for tumor cells that are not recognized. The tumor can also repress immune recognition through multiple means through that selective process. It can lower its antigenic profile, basically favor less antigenic mutations. That basically means that it favors mutations that are not recognized by the immune system. So the immune system of every person has a particular repertoire, and that repertoire is finite and is fixed very early on. And of course, it's constantly adapting to whatever we're exposed with. But if the immune system of that person does not recognize a particular mutation, then that mutation will be tolerated and eventually lead to higher prevalence in the tumor. It also changes its antigen presentation by basically repressing the MHC, the major histocompatibility susceptibility complex, which is where different cells basically present their own identity. So the tumor basically tries to sort of not present itself. And it also establishes an immunosuppressive environment. It basically downregulates cytokine signaling, which basically represses T cell activity. It reprograms the T cells and the dendritic cells in its environment to be less effective. It directly represses T cell effectors by upregulating T cell inhibitory ligands, and it recruits suppressive immune cell types. For example, T regulatory cells are suppressing the immune system, and the tumor basically recruits these cells nearby to help it escape immune surveillance. So this is basically a constant interplay between the tumor and the immune environment. Basically, novel coding mutations in the tumor that help it survive are also generating new antigens that will then lead to the creation of antibodies from the immune system that will then recognize the tumor. So the tumors downregulate their own antigen presentation, they downregulate the immune system, and there may be actually potential therapeutic targets towards this particular process. The immune system, on the other hand, helps select clones that are not recognizing by attacking highly antigenic clones. And then tumor decoy cells can potentially overwhelm the immune system and then let other cells survive. Basically, by proliferating wildly, the tumor might actually simply overwhelm the immune system, leaving some tumor cells behind that can escape recognition. Now, there's recurrent mutations in the HLA genes that actually repress this MHC antigen presentation. So a lot of the HLA pathway genes are very recurrently mutated in tumors. The tumors generate inhibitory ligands that suppress T cell activity. And some immunotherapies are in fact specifically seeking to reverse this effect and in fact promote T cell activity. But if you do that in an unbiased way, you might end up with autoimmune problems. So basically you're trying to sort of boost the immune system to fight the tumor. That might in turn lead to the immune system attacking the body, and you end up with two problems instead of one. And then autoimmune diseases can help trigger immune response against cancer. So again, these problems can in fact cancel each other by triggering this. So here's some of the ligands that actually allow either inhibition or activation of the immune system. These are some of the most common ones. So CTLA4 and PD-1 and PD-L1 are in fact some of these inhibitory signals that the tumor sends to the T cells. And then many immunotherapies are basically trying to block this particular type of signaling in order to avoid the tumor sort of exploiting it to repress the body's defenses. So the tumors generate inhibitory ligands, they suppress T cell activity, and many immunotherapies reverse this effect and promote T cell activity. And more generally, the diversity of neoantigens in the tumor can predict the success of immunotherapy. Namely, the more detectable differences the tumor has that the immune system can recognize, the more likely immune therapies are likely to succeed. So with higher mutational load, you can actually end up with more successful immunotherapy. This is in fact one of the rare occasions where smokers actually perform better because they are actively mutagenizing their own cells. So then when they're in an immunotherapy setting, they end up responding better. You should not smoke regardless. And then there's computational models for actually predicting the neoantigen recognition. So you can predict the likelihood of presentation of different antigens through a particular program that uses a position-specific coding matrix and neural networks to basically look at the sequence of peptides and then predict which immune cells will actually recognize. As I mentioned, there's this dramatic variation in the overall mutation rate of different tumor types. And that also leads to a huge variation, which is very highly correlated in the neoantigen landscape. So the more mutable the tumor, the more antigens it has, and the less mutable, the less antigens it has. And then the heterogeneity within the particular tumor can actually also predict immunotherapy success. Basically, if you're very heterogeneous, that means that the immune system has more chances of recognizing the tumor. And this is, in fact, very heavily dependent on the specific repertoire of your immune cell. So as you all know, VDJ somatic recombination can basically lead to a huge diversity of the recognition profiles of the antibodies that your immune cells can make. And that, in fact, dictates just what you will be able to recognize, A, from your environment, B, from the tumor itself. So these T cells that are infiltrating the tumor are limited in their repertoire. And the specific repertoire that they have dictates whether they will be recognizing the tumor or not. So then the T cell receptor diversity of the specific recombinants that it has led to, the T cell repertoire, is in fact enabling you to now start predicting what sequences you will be able to recognize. You can determine the diversity of your T cell receptors across different cancer types, and then build distinct profiles of your TCR repertoire, and then use the correlation between neoantigens, basically from the tumor side, and T cell receptor sequences to basically start building a huge machine learning problem. It basically says, hey, here's the T cell receptors, and here's the sequences that they recognize. And you can put all of that in a predictive framework that can be either a position weight matrix or a neural net, a deep learning framework that basically allows you to then start building a predictor of which T cell receptors will actually recognize which neoantigens. So then you can detect antigen-specific T cells using MHC multimers. So basically, you can actually build a recognition profile that allows you to do that. And you can use that to start designing specific vaccines that you can then give to a patient based on the tumor of that patient. So the normal way that a vaccine works is that we're saying, hey, let's prepare you for whenever the pathogen arrives. So, many of us take the flu vaccine, and that basically says, well, your body will be ready when the flu arrives. You will already have antibodies for it. Everybody with me here? Now, for the tumor, it's a little different because the tumor is actually evading your immune system. It's already here, but there just simply aren't enough, antigens for your immune system to recognize. So what these personalized vaccines do is that they basically take a sample of the tumor for that person, and then they say, how is that tumor different from the normal of that person? What are the specific antigens that we should be favoring and presenting to the immune system of that patient in order to train the immune system of the patient to then better recognize the tumor? Who's with me on that concept? Awesome, great. So that's sort of what a lot of these immunotherapies are now going after. Basically, they're utilizing patient-specific neoantigen profiles of what are the mutations of that tumor to develop vaccines that can assist the immune system of the patient to fight their own tumors, specialized for the mutations of that tumor. The basic hypothesis is that the new neoantigens will be boosted by, the recognition will be boosted by vaccines, by sort of feeding more of that neoantigen and then promoting anti-tumor response by the T cells of that person. And of course, there's two major challenges. One is, how do you identify both patient-specific and HLA allele-specific peptides? Because the HLA allele of a person will determine which antigens it is more likely to recognize. And also validating that these synthetic peptides indeed assist tumor response in real patients. So this is work by Nirha Cohen and Catherine Wu, again here at the Broad Institute. And what they're doing is that they're predicting HLA allele-specific peptides by, number one, experimentally testing hundreds of thousands of B cells expressing different HLA class 1 alleles. So this is the huge diversity of HLA A, B, and C alleles. You can combinatorially generate the most common of those by synthesizing peptides with ends for every SNP of these HLA alleles. And then creating all of these B cells and then asking which ones respond to different peptides. That allows you to cover part of the space, not all of the space. So then you feed the output of that into a huge neural net, which then allows you to predict for a particular person based on their HLA profile, based on their immune cell profile, what are the peptides that they're the most likely to recognize. So that's part one. Part two is you now take these predictions of what are these peptides that you should be making more of, and then you make them and you insert them into the immune system into the body of these individuals in order to then train their immune system by basically, sort of priming them and then boosting them. And then you look at the response. So in this particular case, they've basically started a clinical trial where they've created these neoantigen vaccines for 16 different patients. And then they've been sort of testing, number one, safety and feasibility, and number two, what is the immune response and what is the progression? And here's the progression over time in months. You can see here a 2-year cycle over which the patients basically receive some priming and then boosting. And you can see here how the 6 different patients responded. And you can see that, nearly all of the patients basically responded quite well. 1, 2, 3, 4 of the patients responded particularly well. And then they verified using both the tumor cells from the patients and wild type cells from the patient, from the same patient, to basically make sure that whatever they fed to the immune system basically trained it to recognize tumor, but not self. And you can see here this dramatic difference in how much more the tumor in red is recognized versus normal cells from the same patient. And now across these patients, basically the 4 of them that responded the strongest have in fact been completely tumor-free for more than 2 years now. And then the 2 that did not respond fully, when subjected to the typical anti-PD-1 immunotherapy that basically represses this repression of the tumor on the on the immune system through this PD-1 receptor that promotes these regulatory cells. Basically, this regular treatment basically led to complete complete therapy in all 6 patients, basically, when you combine the ones that did not respond immediately with this traditional therapy. So this is 100% success compared to a typical 5% success for these immunotherapies for long-term complete lack of new events. So this is, really, really dramatic. And that's when the immune system has these antigens to begin, these antibodies to these neoantigens to begin with. So you're training the immune system by basically selecting the cells that can already recognize the tumor. There's another class of therapy that basically says, well, what if the body doesn't even have anything to recognize the tumor? So what they do here is that they take the blood from the patients, they take out the blood, they construct, they engineer new T cells based on, just a finite supply of these new T cells that will be then able to recognize the tumor. And then they put those back into the patient. I've actually visited the facility where this was developed at UPenn. And it's just an incredible factory where they basically have this, this ability to just take out your own blood or, add the synthetic machines, these T cells, that are completely synthetic that basically will recognize the tumor, put them back into your body, And then they will then go off and fight the tumor. So I just want to remind you that all of these incredibly amazing immunotherapies are just a small part of a whole panoply of different pathways that we're using to fight the tumors. Basically here, this immune aspect. And then all of these different pathways are in fact subject to different types of drugs. So this is an incredible time in the battle against cancer. And I think, you know, the field is wide open in many different ways, basically in being able to exploit all of these different techniques that we learned throughout the class, to basically better understand both coding and non-coding drivers, better understand the gene regulatory rewiring events, and also understand these interaction between tumors and their immune microenvironment. Who feels that they've learned something? Awesome. Yeah, done. Hold on one second.